Hi class, in this lecture, we're going to be looking at genetic recombination. Where we're going is the DNA recombination events. And so this is how bacteria exchange DNA with other bacteria. This is important because when we are talking about the DNA of antibiotic resistance, this can greatly affect the population of bacteria that causing infections. There are three ways that bacteria get new DNA. Conjugation, which is what I term bacterial sex, because basically you have the pillars with bacteria exchange DNA and brings it over. Transformation, in the lab where you take naked DNA out of the cell and add it to the bacteria. They get new genes because of that. Transduction, which is the use of the virus so that the virus infects the bacteria and now gives new genes. We have nice little videos for all these three different ways, but again, we will take a quick look at each of them and show you what I mean about these things. The first one is going to be conjugation. Again, conjugation transfers a plasmid or chromosomal fragment from a donor cell to a recipient cell. Gram-negative cell donor has a fertility plasmid, F plasmid, or effector, and allows the synthesis of a conjugated pillars. Recipient cell is a related species or genus without a fertility plasmid. The donor transfers fertility plasmid through pillars. The difference between donor and the recipient is that one has the DNA and the one does not. What happens is that the bacteria with the DNA can make a pillars. The pillars is the tube that connects to the other bacteria and then exchange the DNA with the recipient bacteria. Think of this as bacterial sex. One bacteria gives DNA to another one. Here is a little video show you how this all happens, and I think this gives you an excellent example of what goes on.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
This shows how the transfer takes place. To remember conjugation, it is the exchange of DNA from one bacteria to another using the pillars. Transformation is something we did in the lab. This is where we are taking DNA from an outside environment and putting it in. Some cells can naturally do this. In the lab, what we did was we artificially made him competent by adding magnesium, calcium chloride, to make it can uptake the DNA into the cell. Some cells are naturally competent that can uptake the DNA and insert it into the host. Now you have added those genes, and so this is called transformation. This is an important tool that we utilize in the lab. This is why it is an essential tool in recombining DNA technology. This is Graves' work, which was the transformation of the string. It showed the idea of what happened. The encapsulated string would kill the mice. The non-encapsulated could not kill the mice. So the mice injected with the heat-killed encapsulated string could survive. However, if you mix those two together, the mouse dies, and the encapsulated string can be isolated from the dead mouse. So what happens is the DNA from the heat-killed encapsulated transformed to the non-encapsulated, that is, transformation. Here is the video to show you how that works.
Transformation is the process by which DNA is introduced into bacteria. Bacteria can then replicate and express that DNA, which can be isolated for downstream applications. Artificial transformation of bacteria involves producing competent cells capable of exogenous DNA uptake. There are two primary methods of artificial transformation used in the laboratory: chemical transformation and electroporation. It is thought that chemical transformation, which requires chemically competent cells, uses divalent cations to increase the permeability of the bacterium's cell wall, thereby increasing the likelihood of DNA acquisition. Many common protocols include a heat shock step to improve DNA uptake. Electroporation involves the application of an electrical field to the competent cells in suspension, thereby introducing holes in the cell's perimeter. The newly compromised cell wall allows exogenous DNA to pass into the cytosol of the bacterium. Outgrowth of bacteria following transformation allows for repair and recovery of the cells in liquid medium without antibiotic. After outgrowth, the culture is spread on selective plates to isolate transformants containing the newly acquired DNA, which conveys antibiotic resistance. A variety of competent E. coli types are available in different formats and for different applications, such as high-efficiency cloning and protein expression. Visit clonewithneb.com for the full list of products available for this application.
This video shows you how you get colony on the plate, because only certain bacteria will get the DNA, and only those that get the DNA will grow. So that is the example here. The only ones that will grow on these ampicillin plates are the ones that get ampicillin resistant. And so this gives you an excellent schematic of how that works.